Electrophoresis and all the other organizers for having me today. Really excited to share this project and this work that I've been working on for the last couple of years. So the title of my talk and this picture on the title slide here might not make any sense to you, but I promise that I will explain what BioBits is, what freeze-dried cell-free technology is, and how this can be applied to classrooms to help students learn about molecular biology and hopefully get them excited about it as well. So where did my mouse go? Oh, there we go. All right, so for this audience, I'm guessing I probably don't need to convince you guys about the importance of STEM education, probably preaching to the choir here. But as you know, trying to get students excited and engaged about STEM is really important, not only just to, get more students into STEM fields, but also in general, just raising the scientific literacy in our country. I like showing this graph a lot from AAAS, where they did a survey of general adults in the US versus scientists, and noticed this huge gap in just general biomedical sciences knowledge. Things like, is it safe to eat GMOs? Like, there's a 51-point gap between scientists and non-scientists. Things about evolution, things about vaccines, and more timely with the pandemic going on, there's all these myths and misconceptions floating around about the COVID-19 vaccine and everything. So it's not just about getting more students into STEM fields, it's about just generally better educating these students, so even if they grow up and they don't go into a STEM field, hopefully they still have retained some of this knowledge about how basic biology works, and so they can better understand these ideas, right, like vaccines, when it is applicable to them. So switching tacks, I know not everyone here has a background in biology or biotech. Biotechnology in general, if you're defining it at a very high level, is the idea of using living systems to make products. So the earliest example of biotechnology is agriculture, right? We are growing these plants in order to make food and other materials to support humans. And farmers would crossbreed plants to grow hardier plants, and over time we have crops that can survive droughts and bugs and all that kind of thing. Other examples of early biotechnology is the idea of fermentation. Ideas that help us make beer and wine and bread. But when you get down to the molecular biology level, we can get a lot more detailed. And once humans understood what DNA was and how genes work, we were able to actually use living systems to make more specific things like medicines. So a very common thing done in the biotech fields and the pharmaceutical companies right now is taking DNA that encodes for proteins that could be used for medicine. A commonly used example is insulin, right? So you take the DNA that encodes for the insulin protein and you stick it into bacteria, and this bacteria becomes your factory or your workhorse. The bacteria takes the DNA, reads the instructions, encode it in the DNA, and is able to make insulin, which then we can harvest and use to treat diabetes patients. So this is a very common technique used in pharmaceutical companies in order to make protein-based products. So medicines, but also things like biofuels and other biochemicals can be made using mostly bacteria, but other cells as well as your workhorse. So kind of teaching this, going back to the classroom, teaching this at the middle school or the high school levels is really important. Understanding what DNA is, understanding how DNA is used to make proteins, all these really basic ideas. And there are actually quite a few hands-on molecular biology activities out there that are commonly used in classrooms to teach students this. One example is doing bacteria transformation, which is the process of the bacteria uptaking the DNA. So in this experiment, students are taking bacteria and transforming them with DNA that encodes for a green fluorescent protein. So that makes their bacteria glow green as shown in this plate here. So this teaches them not only the techniques behind, these important biotechnology techniques, it's also teaching them, what DNA is and how it's used to encode protein, and how, scientists and researchers can harness that ability to make the proteins they want. Other techniques include gel electrophoresis. For those of you who haven't done gel electrophoresis before, this is a method of separating DNA on what looks like a little slab of jello, and it's really informative in kind of demonstrating a lot of other ideas in biotechnology. So these are very commonly done activities right now at the middle and high school level, but there's a lot of drawbacks to this, mostly because these labs can be really difficult to implement in classrooms, especially classrooms that don't have a lot of resources. Generally, molecular biology can be pretty hard to understand what is happening. As cool as your endpoint experiment might be, the process of doing it can be boring to students. If you've done molecular biology labs before, it's honestly just a lot of mixing clear liquids into other clear liquids and hoping magic happens. And so for a student learning about these basic molecular concepts for the first time, they might not get what's happening because they just see clear tubes like this. It can require really expensive equipment, things like freezers, like -80 Celsius freezers to store your reagent, things like incubators and shakers in order to grow up these bacteria cells to use in your classroom. Overall, it requires a lot of time and expertise that a lot of teachers don't have, especially teachers and schools that might not have a lot of resources, right? And like Justice said, I really believe in making science as accessible as possible. And I don't think it's fair that it's just, the rich private schools that are able to do these labs in their classroom. Part of my motivation for doing my thesis was to find a way to make it more accessible, that so schools that don't have expensive equipment, schools that don't have teachers with biotechnology training still could use these activities in their classroom to teach their students about these basic concepts. So that's where the bulk of my thesis comes in, which is this idea of cell-free protein synthesis. And in the end, if this doesn't make sense, feel free to ask me at the end of my talk to clarify this. But just really briefly, what cell-free protein synthesis is boils down to me taking the bacteria. So again, this is the bacteria that we can use as our workhorse or factory to produce the proteins we want. Taking this bacteria, and instead of using the live bacteria as my factory or workhorse, I bust open these bacteria cells and scoop out the stuff inside. So scoop out the molecular machinery that's inside, things like RNA polymerases, things like ribosomes and tRNAs. Put that in a little tube. Make sure there's other things in the tube like nucleotides, amino acids, and energy sources. And if you're not familiar with these terms, these are all of the molecular machinery and kind of ingredients involved in taking DNA to protein through a process called, there's transcription and translation. It's the whole central dogma of molecular biology. But basically, going from the instructions found in your DNA all the way to making your protein requires this molecular machinery, which is typically found in live bacteria cells. But a long time ago, scientists found out that you could just bust these cells open, scoop out the innards, and just use that as a static reaction in a test tube. And in this way, it's abiotic, meaning that it's not alive. You don't have to worry about keeping cells alive, so you don't need incubators and shakers and all that expensive equipment. And it's also an open reaction because it's not kind of blocked by bacteria cell walls or cell membranes. It's a lot easier to get in there and change variables and mess around with the system inside, which is really great for hands-on learning. And so I joined Jim Collins's lab at MIT during my PhD, and they were doing a lot of cell-free protein synthesis. And I'll get into some of the examples of really cool applications of this that are non-educational. But I looked at this and realized, hey, we could probably apply this to education. Oh, and quickly, this is the analogy I like using to explain cell-free protein synthesis a little further. Imagine you have a car and you pop the hood of that car and you grab the engine inside. you can hook that engine up to something and still have it run, right? The engine has no idea that it's not in the car anymore. And it's kind of the same thing with the bacteria, right? You can pop open the bacteria and scoop out the machinery, the engine that's inside. And again, those molecular machinery parts don't know they're not in a live cell anymore. And as long as you give it the right ingredients, the right reaction conditions, it will still do what it's supposed to do, which is taking DNA and turning it, using those instructions to build and assemble proteins. And one innovation we did in the Collins lab is to make these reactions last even longer. So if you put all those ingredients in a tube, you still need to keep it on ice, and you still probably need those fancy -80 freezers. So to get around that, we freeze dry these reactions. And freeze drying is a process where, let's say you have an ice cream cone. If you just left this ice cream cone out in the sun, it's going to melt. What we can do to this ice cream cone is you can dunk it in liquid nitrogen and then put it in a special machine called a lyophilizer or a freeze dryer, which sucks all the liquid out of that frozen object and just leaves the solid chunk behind. So if you ever had astronaut ice cream before, this is what it is. So basically, we did the same thing to these reactions. We dunked them in liquid nitrogen so they froze, and then we stuck them on the freeze dryer to suck out all the water. And what we were left with these freeze dried cell-free pellets. So now these pellets, circling it there so you can see, these pellets make shipping and storing so much easier. We can actually ship these at room temperature. We don't have to worry about keeping them at like ultra low temperatures or anything. If you've been kind of following the news right now, right, like mRNA vaccines, one of the difficulty in distributing those is that you have to keep them at super low temperatures. And that is the case for a lot of shipping molecular biology reagents in general. But by freeze drying these reactions, we're able to ship at room temperature. And once the teacher or the classroom receives them, you can just store these in a regular fridge or freezer. They even last at room temperature for a couple of weeks. But for longer storage, you can put them in the regular fridge, regular freezer. No fancy -80 or ultra low freezers needed that are really expensive. So yeah, so that's cell-free protein synthesis. And when I joined Jim Collins' lab at MIT, they were applying the idea of freeze-dried cell-free protein synthesis to a lot of really, really cool applications. Things like being able to make medicine and vaccines on demand, right? So basically in those tubes, you have a little protein factory. And as long as you give it the DNA that encodes for the protein you want, theoretically, you could kind of take this anywhere. And here's a little mock-up of a kit they designed in the Collins lab, where this idea is you have a little, almost like a little briefcase full of these freeze-dried cell-free reaction, like kind of a library of DNA that encode for the things you want to make. And, depending on where you are and what you need, you could make the medicines and say the vaccines when you need it, right at the point of care. So you don't have to worry about transporting it. You don't have to worry about making too much and it's not needed. You just make it when it's needed, which is a really cool kind of theoretical application of freeze-dried cell-free technology. It can also be used with biosensors in order to detect different things. So here's an example from one of our collaborator labs, building a biosensor that you can put into the cell-free reaction that detects water quality. So these biosensors are able to detect things like antibiotics, chemicals, and metals. And I'm not going to get into the details here, but basically, if any of these things bind to the biosensor, chemical reaction or biochemical reaction happens using the cell-free system to create an enzyme. So it's using the cell-free system to make this enzyme protein, and this enzyme will convert a colorless substrate into a yellow substance. So it's a colorimetric output. If you see your thing turn yellow, you know that your sample of water that you add in has the antibiotic or the chemical or the metal or whatever your biosensor is supposed to detect. So that's really cool. And this can even be leveled up into diagnostics for human health. So this was a huge project done in the Collins lab as well, where, back during the Zika virus crisis, they developed a sensor, a biosensor that's able to detect whether or not a sample has Zika virus or not. And if it does, these little yellow discs will turn purple as a really quick, rapid, low-cost way of diagnosing patients. And especially, there are a lot of like developing countries that don't have access to good health infrastructure or whatnot. And so having something that could be done very quickly with not a lot of expertise or medical infrastructure would be really helpful. So this is kind of our idea of how to use freeze-dried cell-free. So in this case, the reactions are freeze-dried directly on these little paper discs shown here. And so when I joined the Collins Lab, I had a background in, outreach and volunteering for a lot of STEM initiatives. And I looked at this stuff and said, okay, they're using it for diagnostics because it's cheap, it's easy to use, it doesn't require a lot of equipment. And I was like, well, it sounds like we could apply this to education, right? Classrooms and teachers are always looking for things that are cheap and easy to use, but still can demonstrate a lot of really great hands-on molecular concepts. And so this schematic is just kind of showing our ideas of how we can apply these freeze-dried cell-free reactions to education. This idea of you have your cell-free reaction here, and all you have to do is add the DNA and water to rehydrate this, maybe some optional substrate molecules, and we could produce a bunch of different proteins for different educational demonstrations. We prototyped a lot of different activities just to see what would work, ranging from things that you would see visually, like fluorescent proteins and colorimetric proteins, things like enzymes that could generate smells like banana, other enzymes that are able to cross-link hydrogels that students can poke, and even kind of getting into introductory synthetic biology with programmable biosensors that students could play with that gets into the kind of diagnostic field. So over the course of my PhD, I prototyped a huge wide range of different activities. And this was just kind of just seeing like what works in this system that could be potentially used to teach different concepts. If you're curious about some of these prototypes, we published 2 papers in Science Advances in August of 2018, as well as another paper in ACS and Bio in March of 2019. Both made the cover, which is really cool. And both of these, all 3 of these papers are open source. So if you wanted to check them out, you don't have to worry about the paywall. And for me, I found that the fluorescent proteins were really, really fun. They were very robust. You could easily just visualize them. I'm showing what I use at Mini PCR Bio now, but we actually prototyped our own acrylic version of this. Basically, this is just a handheld blue light illuminator. You shine the blue light on your tubes, and if there's any fluorescent proteins in there, they will glow. And we found out that, you could really, really robustly express these fluorescent proteins in the cell-free system. And kids get really, really excited when they turn this thing on and they see they have bright colors. In fact, we're making so much protein that we don't even need the blue light. Sometimes you can just hold it up to a piece of paper and you can see the different colors you're making. But I think they look cooler under the blue light. So using this system, this could range from, things like basic gene expression and teaching protein synthesis. So in this activity, we actually went to a high school and gave the students a 96 well plate that all of these little holes here contained a freeze-dried cell-free reaction. We gave them all the DNA and, taught them basic gene expression and, how basic gene expression and protein synthesis works. And we let them design their own picture here. So they were like, okay, if I want the red fluorescent protein, what is the concentration I need of this DNA to add to this reaction? And they planned it out on paper, and then they actually kind of executed it. So it was a really great example of, I forget the build, design, and test cycle. And so these pictures are actual designs that kids made, which I think is really cool. But you can go very basic like this, all the way up to getting into biosensors and diagnostics. So this is an activity that I prototyped where, I don't know if you guys have done the strawberry DNA extraction where you mash up a strawberry and then extract the DNA out with alcohol. We did the same thing, except we used a banana or a kiwi. And once we got to the DNA, this is usually where you stop with the DNA extraction. Usually you just kind of stop and see the goopy DNA and you're like, okay, that's really cool. But in our activity, we actually went a little further. We actually amplified up, we had students amplify up the DNA and then stick it on one of our sensors. And I built a sensor that actually can tell whether or not it's banana DNA versus kiwi or strawberry DNA. And I built one for kiwi as well. So if it senses banana DNA, it turns a green fluorescence, which is pretty cool. So we prototyped all those different activities. And then towards the end of my PhD, I, decided I really wanted to make this into a real thing that students and teachers could get their hands on, not just the prototypes that I had in the lab. And so for the first activity, through talking with teachers and interviewing them and kind of seeing like what was missing in terms of a hands-on activity in their classroom, I decided to develop a activity that covered the central dogma of molecular biology. So the central dogma of molecular biology essentially represents the flow of genetic information from DNA to RNA to protein. So when DNA gets copied over into RNA, that information gets copied over to RNA. It's a process known as transcription. And when it gets when that information then gets translated over to making protein, that process is known as translation. Sorry, the lighting here on my webcam is really weird, so I'm kind of in the shadow. So please excuse me for that. And so we wanted to build an activity using the cell-free system that demonstrated this. Because this, to students looking at this diagram for the first time, just seeing all these squiggles, it's, very abstract to them. And then, it's something that they can't see. So it might be really hard for them to wrap their head around. So I cloned a DNA plasmid that allows students to visualize both RNA and protein. And the way they're able to visualize RNA is I built into the gene something called an aptamer. And just briefly, the way it works is when DNA goes to RNA, you have your RNA. This aptamer sequence here in the RNA will actually fold up on itself and form a little binding pocket, which will bind to a chemical. And once this binding interaction happens, it emits a green fluorescence. So when you see green, you know that you have RNA and that transcription must have been successful. So students can actually, in real time, looking at their experiments, see RNA get made, understand transcription. And kind of similarly for visualizing protein, we just use red fluorescent protein again. So a red fluorescent protein, which fluorescent proteins are actually found naturally in things like jellyfish and coral. So we use the red fluorescent protein that's naturally found in coral, got the protein coding sequence for it. And so in your BioBits system, once you go through both transcription and translation, you end up with a bunch of red fluorescent proteins. So once students see the red, they understand that they have protein now and that translation was successful. And so this video will play. You can actually see it go from green and slowly transition to red as translation happens, which is pretty cool. And so students are actually able to visually see DNA go to RNA, go to protein, understand what is needed at each of those steps, and understand how those steps are different and how they are related. I particularly like this activity because it highlights the middle step. I always joke that RNA is the forgotten middle child of the central dogma. Everyone talks about DNA, everyone talks about protein, and no one really knows what RNA is, as evidenced by, the new mRNA vaccines that are being distributed right now. Everyone was like, what is RNA? Is that a weird kind of DNA? And so I like the fact that this activity really shows you, what RNA is and why it's an important part of the whole process. All right, so at the end of my PhD, finished at my thesis, I decided to send this activity that I developed, the central dogma activity I developed, out to 50 schools across the US just to pilot test it in those classrooms and gather feedback. And I'll just caveat right now, I don't have a background in education or educational assessment. So a lot of the feedback I gather, some was quantitative, but a lot of for me was just kind of qualitative, asking teachers and students how it went. But for example, I asked teachers what was their favorite thing about the lab. A lot of teachers said, really appreciated how easy it was to use, how easy it was to set up, how little equipment was needed. Teachers also commented on the educational value of the activity. And the thing that really, really jumped out to me was teachers saying that students felt like scientists and doctors by, just like touching and using the laboratory materials. And I feel that self-identification with being a scientist is so important at that age. So that really jumped out at me. And yeah, also a lot of teachers saying like the lab was reliable, which apparently doesn't always happen with biology. If you've ever worked in a biology lab or molecular biology lab, you know that biology labs always almost fail. So, but for students, we were trying to make this as robust as possible. I did measure student learning as well. I'm not going to get into this here, but I did kind of quiz them before and after running this lab, how much they understand about DNA, how much they understand about proteins. I did see a significant increase in their learning before and after they did the lab. Obviously, there were some things that I learned that could be improved. The biggest thing was kind of the curriculum that went along with the lab, right? It may be fun for the students to pipette and everything, but if there's no guiding curriculum that really walks the students through what is going on, they're not going to really take that much from the activity. And again, if they're not guided through the curriculum, again, they still might not understand entirely what's going on. And so once I graduated from my PhD, I took all the BioBits work and went to Mini PCR Bio, which is a small biotech company that's based out of Boston. So I didn't have to move anywhere. But they had a similar mission statement to me, which is the idea of making science accessible for anyone, anywhere. They're called Mini PCR because their first product was a miniature PCR thermocycle. Hold on, mine is sitting under my desk right now. This thing, basically being able to do PCR anywhere. And they were looking for kind of like a portable protein synthesis platform, which is what I had with BioBits. So it was a very natural partnership. We were very much aligned on our mission and values in terms of how we're going to use this technology. And plus they had a lot of experience in developing not just the technology, but the curriculum as well. And that was the piece I was missing that I learned from my feedback from the pilot study. So after joining Mini PCR, I developed a really thorough accompanying curriculum to go along with the central Dogma activity. I made it highly visual, I made it very self-inclusive, and yeah, took it from prototype to a real lab activity, going through product design and production, which I got to oversee, which is really cool. Launching it and seeing it being adopted into classrooms, actually getting to go to classrooms and see teachers use this to teach to students was really, really cool. And even over the last year, adapting this lab to remote learning, and we actually made a at-home version of this kit where teachers could send each of their students the kit and do it all over together over Zoom. Since it's a very visual lab, right? The teacher could just demo it and hold up the results of the webcam, or it's it is affordable enough that some schools were able to get the funding to send out individual components to everyone to do, which is really, really cool to see. And it's not just limited to the US. BioBits has been taken all around the world, which I am thrilled about. Here's a picture of it being used in Japan to teach a teacher workshop there. People have taken my curriculum and translated it. This is German and a Turkish translation of my curriculum. And we're even launching this to space next year, which is insane to me that it's going to space. But we at Mini PCR also run a science competition called Genes in Space, where students propose molecular biology experiments that astronaut scientists can do on the International Space Station. Winning experiment goes to space, and for next year's this year's contest cycle being launched next year, students get to use BioBits as part of their experiments. So I'm very, very excited to launch my little BioBit babies to space next year. And obviously there are a lot more BioBits labs to be developed besides the central Dogma. We just launched one on protein structure and function, teaching students about primary, secondary, tertiary, and so on, structure in proteins, and how protein engineers can take advantage of that knowledge to engineer their own proteins. And just previewing it now, I'm also developing one on antibiotic resistance to demonstrate, how antibiotics work, how resistance can arise, and why that is a problem. So I'm using BioBits to do that as well. So yeah, just in summary, through the course of my PhD, I realized that cell-free tech and these genetic sensors could be used to develop a lot of different hands-on activities. And because it's easy to use, and because it's more accessible, and because they have really visual outputs, we were able to develop activities that really increase student engagement and connect it to real world ideas. And like I said before, because it's cheap and easy to use, we made it more accessible to more classrooms. And one thing I always like asking every time I present this is kind of to think about your own research and how that could be translated into an educational application. Every time I give this talk, I always get someone coming up to me and asking, oh, I do XYZ in the lab. How can I turn that into a fun hands-on activity for students? Just kind of like thinking about that is always really a great discussion point with people. Yeah, just quickly wrap up with acknowledgments. So I did this work at Jim Collins's lab at MIT with a lot support from my lab mates. We also collaborated heavily with Northwestern University, the Jewett lab there. Jess Stark is my co-developer for BioBits. We worked on this together. And then the Weiss Institute as part of Jim Collins's lab also was a huge support in this. And then the mini PCR bio team for helping me turn this from a prototype into an actual product. So yeah, that is everything. I'm happy to answer any questions. I'm happy to dive into more of my own career, PhD side of things, doing a thesis. Happy to talk about, the more company side of things with mini PCR. Yeah, feel free to ask away.
Thanks so much, Ali. That was, I'm sure I speak for most, if not all, was such an innovative and sort of refreshing sort of insight in life science specifically. And so selfishly, if it's okay with everyone else, I will ask the first question because I'm sort of dying to know, but if there are others, you can put it in the chat or jump in at any time. But one of the things I think is really interesting is sort of a life science teacher and really advocate. I really like how you sort of frame this, or at least one of the needs for this around infrastructure, right? Because we know that in order to do really sophisticated things in life science, or at least innovative things, they're very costly. And I can't help but remember when personal computers were, I'm old enough to remember that personal computers weren't always personal, right? And to be able to get those in the hands of learners became such a sort of game changing and consequential innovation for computer science. And we still have a lot more to do there. But in life sciences, I see there being a sort of a similar parallel. And so since your work sort of exists on the very edge of these fields, I wonder if you could talk about maybe two things. One is, where do you see the direction headed in this area? I think a lot about designing with biology, and I wonder where you see there being overlaps. And then the second one, and maybe I should have asked in reverse order, is maybe, and you alluded to it, if you could talk a little bit about, and I'm asking this question because I know there are students here who are thinking about their own classrooms as they transition into teachers, becoming practitioners, and some of them are thinking about continuing science. And so can you talk a little bit about not only where you see the future of this, but also how you sort of got started and your trajectory into, or foray into, this field as a budding student, scholar, teacher, and now innovator?
Yeah, for sure. All right, so I'll answer first kind of my career trajectory, I guess. So in developing the BioBit system, um, when I first joined Jim's lab at MIT, after reading all these articles about all the really cool freeze-dried cell-free technology he was developing for diagnostics in that lab, and me just, with my background in STEM outreach, and volunteering, I was just like, well, we could really just apply this to education. And when I first started the thesis, I wasn't entirely sure where that would take me. It was just kind of like, well, let's just try to do this and see if it works. And from there, I just kind of kept building and building. Like, okay, wow, we prototyped this and it worked. What happens if we stick it in the hands of real teachers and real students? Will it still work, or does it just only work on my lab bench? And that's where the pilot test came in, and got a lot of great feedback from that. And so at that point, I was towards the end of my PhD, trying to figure out what I was going to do next, and getting a lot of this great feedback from teachers. After publishing those papers, I got so many emails from teachers asking, like, if this is a real product they could buy. And I had to tell them, no, this is only a prototype. But the more of those emails I got and the more feedback I got from the pilot study being like, this is great, we want to use more of this, started thinking more about, hey, maybe we can actually turn this into a real thing that teachers and students can get their hands on. And so I remember towards the end of my PhD, I was talking to so many different nonprofit organizations, educational organizations, basically just trying to find a home for BioBits, even considered creating my own startup around this. And kind of what I learned from just talking to everyone in this was that for me, ownership of BioBits was never that important to me. I didn't need like a patent on it. I didn't need to like make millions off of this. For me, the important thing was being able to get this out into the world to teachers and students for as quickly and as cheaply as possible. I didn't want to reinvent the wheel by starting my own company. I wanted to take this to an organization that already had that infrastructure in place and knew how to write curriculum, knew how to produce, and knew how to distribute. And so it was really fortuitous that I met the guys at Mini PCR Bio because, a lot of their previous curriculum that up to that point had been all DNA based, and they were looking for something that was very protein based. And so it was just kind of perfect timing that we met and realized that we were just going to be a perfect match together. So I could bring BioBits over to them, be able to, use their existing infrastructure. For example, like I had built my own little acrylic fluorescent imager, and I was like wondering how I was going to scale that up. And then when I talked to BCR Bio, they already had a little cardboard one already in production for some of their other labs, and I was like, well, that's perfect. We can use that for BioBits as well. And so it was really just kind of a perfect match. And they are, and the most important thing was like their mission was just like mine. The idea of it's students and teachers first, right? Like we're a company second, but first it's like just the teachers and the students and the customers. And so I felt very comfortable taking my research and taking my BioBits over to them to, make this into a real product. And moving forward, we're launching more just like kind of generic, like I call these more kind of like canned labs where it guides you through like step one, do this, step two, do this. And I feel like those are really important for students learning about it for the first time. But where this is going, definitely when we first started BioBits, kind of our eventual dream was to have this kind of open source sharing network of biologists and like biomakers and people being like, hey, I created this DNA plasmid that can express this really cool fluorescent protein. Here's a sequence. Anyone can use it. And like a cell-free system, just sharing all these different tools and ideas, exchanging this information, kind of like a giant like a library database, I think is like the eventual direction we want to take BioBits. Right now, it's just very much like, all right, let's teach the students like very basic things. But then, we're talking with people at iGEM, we're talking with, like, we work closely with BioBuilder all the time, being able to take it from just like, like you said, Justice, like inquiry-based learning to students having kind of open design. And we already have students who are just buying the Central Dogma kit as is, and just instead of following the protocol we set up for them, just using it for their own research as well. And so being able to kind of tap into the more, I guess, like sandbox arena would be where we want to go next.
That's great. Are there any other questions from the audience at this point?
No, sir, you kind of took mine. I was going to ask her what were her future plans for, what was something new that she was trying to develop, but I guess you already answered that question.
Sorry.
Yeah.
Yeah, and other things like the papers that are linked in the chat, like, we're definitely not abandoning all those prototypes. Some of those prototypes we have learned don't work as well in the classroom setting. As cool as the smells output is, that's not very robust. So we're probably sticking with fluorescent proteins for now. But there are a lot of other prototypes that are in those papers that'd be really cool if we could figure out a way to make them robust and translate them into the classroom.
Ali, even though that you say that you don't have experience being an educator. The way you explain things, I think it's just a model for someone coming from a pure science background and then making it so visual, simple, and understandable. I think that this is a place where I beg your explanation, perhaps with the COVID situation and the vaccine, and everybody got vaccinated. So in your own words, how mRNA is different from regular RNA, and why some producers use this model as opposed to other producers of vaccines using other models.
Yeah, that's a very timely topic. And in fact, we just released, in addition to our Central Dogma Lab, we actually released a little student guide add-on for teachers to recontextualize the results of the Central Dogma Lab to explain to their students how mRNA vaccines work. So I've been doing a lot of thinking about this. But basically, so there are a lot of misconceptions around what mRNA even is. And so, like I said, this Central Dogma Lab demonstrates that it is the bridge between DNA to protein. So in the Central Dogma Lab, you see your tube go from blank to green to red. And so the way I explain it to the students is, okay, so that step where it goes from blank to green, that is a step of DNA going to RNA through the process of transcription. And that visual step you're seeing in your lab results represents how biotech companies like Pfizer or Moderna might use to produce these mRNA vaccines, right? They're using DNA and they're actually using in vitro transcription reactions to generate a lot of RNA. So they're only doing that step. So when you see blank to green, you are seeing, what the biotech companies do. Now, the second part of your lab results is seeing then your green tube eventually turns red. And that is representing the RNA, the genetic information found in the RNA is being translated into protein. And so that visual step represents what happens after the mRNA is injected into you, right? Your body is taking that genetic information from RNA and making the viral protein, the spike protein that is part of the virus for your immune system to recognize. So traditional vaccines, I'm not entirely sure if it's like the Johnson & Johnson vaccine, but traditional vaccines often just use that last step. They often just make the protein aspect and have that injected into you for your body to recognize. But that's a little harder, right? Because you have to go through two steps. You have to go through transcription and translation. It's a lot easier for companies just to do transcription and then you inject that and make your body do the next step, translation. So for a thing like a pandemic where we really need to get lots of vaccines really quickly, being able just to do the transcription step allows companies like Moderna and Pfizer to scale up a lot easier. And it also gives the advantage of if they need to change if they need to slightly change the genetic information because of variants and whatnot, they can more easily, they're more flexible to switch versus it's a lot harder to go all the way to protein purified and then realize you have to start over because of variants. So that is the huge advantage of the mRNA vaccines. The Central Dogma Lab even demonstrates how, why it's important for the mRNA vaccine to be kept cold, because students, after they come back the next day, they'll see that their green signal is gone because their mRNA in their reactor has broken down in their reaction, which is similar to what happens to, mRNA in the vaccines that are not kept cold. It breaks down, and it's another way of just showing like the mRNA breaks down so easily, it's impossible for it to change your DNA. And also showing the Central Dogma, DNA to RNA to protein is a unidirectional flow of genetic information. So it's not going to go back and change your DNA. So being able to use all of these different visual results they get from the lab to explain how these vaccines work and also explain why some of these misconceptions are false. We've been doing a lot of thinking about this. That wasn't just off the cuff. And yeah, definitely the way to me learning how to explain this to students learning about the first time was definitely a skill that I learned after joining mini PCR Bio. It wasn't something that I had going in. It's a year, almost 2 years now, of learning how to communicate science, and especially this last year, the pandemic, just hosting lots of webinars explaining my science to students, and just learning how to explain it in a way where it makes sense to someone without much of the background.
Hi, Ali, this is William. And thank you for your presentation. It was really interesting and illuminating on the sort of what we, the science teachers, can do in the classroom with the students. I have a comment and a question.
Mm-hmm.
And following on what Murad just mentioned about the COVID vaccine, I think this material, this instructional resource, the kit, is really interesting because of a few reasons. First, it is timely, as you mentioned, because of the current pandemic. One thing I remember from different studies and from my own practice, asking elementary and high school students about their perceptions or views or images about scientists, and many of them draw or describe scientists or the work they do as finding cures to illnesses. That's a big thing, that a big perception or descriptor students use to refer to scientists. And this is interesting because of the implications for understanding what science really is and how it works. Even they say that scientists are involved in the production of new medications. They seem, the students, to perceive scientists as not having problems at all, going from A to B and C and finding cures and making it available to the public. With this kit in the classroom, the students and the teachers will be able to make these molecular concepts many times abstract to them, visible to the classroom, as you mentioned, with the fluorescent light. But also they will spark a conversation about the history and nature of science, and that science has some limitations. That, for instance, if we look at the race to produce the vaccines, it is not that simple or easy. Yeah. Even we have some pharmaceutical companies dropping out of the race, like Merck, because of some issues that happened there. So this is a great opportunity to promote scientific literacy, which is a big component of science education globally. Scientific literacy in connection to, this is an absolute bulletproof solution. Is that what scientists do? What about things like some people are not really willing to, or are hesitant to get vaccinated because they have some beliefs. So that is tied to science education. After so many years of schooling in science, we have these perceptions about using the products of science. So that's one thing, that it is really timely and very appropriate for discussing these socio-scientific issues in science classrooms. My question, so does this kit or the BioBits allow the students to sort of test their own ideas about taking this material or resource in a different direction? What if they come up with their own sort of little simple experimental designs that they want to carry out using this kit? Can they use it to test their own hunches?
Yeah, and like I said before, that's kind of the direction we want to eventually take this, right? From the inquiry-based, the Central Dogma Lab is very much inquiry-based lab, where it tells you this is what you do, this is what you discover. But yeah, being able to take that and give students kind of free reign over the material. And we already even have teachers and students purchasing the regular BioBits labs and just kind of tossing the plasmids that come with it and just use the BioBits system and use their own plasmids and test new things. I always hear I get teachers emailing me all the time telling me about the really fun experiments their students have done. One example I love saying is like one group of students were, discussing, DNA stability and RNA stability, and they said, hey, there are enzymes out there that break down DNA and RNA. I bet that those enzymes are found in like our saliva. Let's spike our saliva, our spit, into these reactions. And turns out if you spike your spit into these reactions, it kills the reaction completely and doesn't work. And so I've had students put the DNA under UV light for a certain amount of time to model DNA damage. I've had students adjust the pH, adjust like kind of the salt concentration, add alcohol, that kind of thing, just to kind of explore what they could do in the system. And I've also had a lot of students kind of build, especially in kind of the iGEM world, build their own sensors and kind of use BioBits as a way to quickly prototype and screen some of their sensors. I've seen that as well. And so right now that's all driven by teachers and students. We don't really have many resources to guide students for that, but that's something we definitely kind of want to move towards. I think I see a question in the chat. Someone's asking, in using these, okay, very interested in using those educational kits for remote learning. I wonder when students perform those experiments in different places, what would that matter? Yeah, so that is one thing, like whenever the big challenge of turning a prototype into a product is to test how robust it is, right? Students, especially students who are using a micropipette for the first time, are often going to add too much or too little, right? So you want to make sure that your lab is set up in a way that even the student messes up a little bit, they will still get results. So we do look at things like if they pipette too much, if they pipette too little. We do look at things if their temperature is slightly different, things like humidity and sunlight. And so we try to write the protocol as detailed as possible. Things like, hey, when you're incubating reactions, like don't put it in direct sunlight, like put it in a dark place. But we've found that at least for these reactions that so far that we've been doing, it's pretty robust in terms of ranges of temperatures. It's pretty robust in terms of ranges in humidity. I've done a lot of these workshops with students and teachers over Zoom. And yes, you do see a lot of variation. Most of the variation is due to pipetting, micropipetting. But for the most part, even if a student adds twice as much as they're supposed to, or twice as little they're supposed to, we do see that they still get some results and are still able to see it. And that kind of goes back to like demonstrating that science isn't the easiest thing in the world, right? Obviously, we want a product that is robust and works like we're very, very reliably. But we can make the most robust product. There's always going to be a student who is creative enough to break that product. And so we always encourage teachers in that case to kind of, turn that, don't frame it as a failure, but frame it as a learning opportunity, right? Like, why did your experiment fail? What controls do we have in place in our experiment so we can actually rule out these reasons and kind of focus on these reasons. And so, yeah, we're trying to make it as reliable as possible. But we're also trying to encourage teachers in the event the experiment doesn't work to kind of frame it scientifically and kind of explain to the student what's going on.
Ali, I really, your answers are so, I'd have to agree with what others have said. They're so straightforward and practical. We've reached time here, and so I'll close by thanking you a whole lot for taking some time to meet with us and to share your work. I thought your trajectory and your story is so fitting, especially for a lot of the pre-service students here who might be thinking about pursuing their PhD here at the College of Education. I learned recently there's a BioBuilder team in El Paso, and I met with the teacher. And so it's starting to flourish in the region. And so if there are teachers here who are interested in pursuing this further, I think your message is it's doable and you can even make it work for your career. And so thank you, Ali.
And I put my email in the chat in case anyone wants to reach out to me and ask about specific things about my career or specific applications of BioBits or anything. Just feel free to email. It's in the chat now. Just ali@minipcr.com.
Fantastic. Thank you, Ali, on behalf of myself and STEMers and the faculty at the College of Education and at UTEP more broadly.
Yeah, thanks again for having me. This was fun.
Take care.
All right, take care. Good night, everyone.